Yeah, so in this session there was 4 talks, and there was this different aspects of studying neuromuscular disorders in the dish. So for example, my talk focused on patients with FKRP mutations using just a monolayer, using a cocktail that induces the maturation and discovering additional mechanisms underlying the disease. And then there were talks from Penny Gilbert studying Duchenne muscular dystrophy and the neuromuscular junction in vitro, which is impossible to study using samples from patients, and discovering new components that are important for disease pathogenesis and potentially find new therapeutic interventions. And the same coming from Saverio Tedesco and David Mack. They were modeling using 3D structures so you can really use patient samples and starting to recreate the muscle in the dish, which really allows to study, again, disease mechanisms that, harder to do in an animal model. It's not quite the same as studying the patient sample in the dish. And once you have a robust model, as we have been witnessing a lot of progress, you can start asking questions in terms of potential new drugs and testing current treatments like gene therapy, as Saverio showed yesterday. I think this aspect of maturation is still a very important one. The investigators have been showing that 3D is still not probably very applicable to many investigators who have the access to perform such studies. So finding more or different approaches to generate a more mature muscle in the dish that investigators that don't have the accessibility to do 3D structures might be able to also be contributing in this area, will be desirable. And I think one aspect that it would be important is just having more access to samples from patients with rare diseases. Because for example, I was working with a sample that's a very rare disease. I had a sample from one patient. So you want to increase our N, and sometimes it's very difficult to reach out to fibroblast samples, because with iPS cells, as we generated induced pluripotent stem cells from any material, right? So if you have a bank, biorepository, fibroblasts, or even blood cells, you can reprogram the cells to iPS, and now you can study muscle from those patients, right? So I think if you can increase this knowledge and have more access to samples from patients from fibroblasts even, we'll have more material to study these rare diseases. Induced pluripotent stem cells, you can specify the cells towards a specific lineage. So when you're modeling the cells in the dish, we are making myotubes. So the myotube is the equivalent of a myofiber in a person. So this is like the final cell, but prior to generating these myotubes, we actually generate large numbers of myogenic progenitors. So my lab has established this many years ago, and during the last 5-6 years, we have been working towards testing the clinical application of these cells after many years of work. So we have demonstrated in many studies that these cells, once you transplant in animal models of muscular dystrophy, including Duchenne muscular dystrophy, limb-girdle muscular dystrophy, FSHD, we are able to generate healthy muscle that's functional, and the cells are able also to seed the stem cell pool, which is very important for long-term regeneration. So after all this extensive proof of principle, we have recently generated clinical-grade myogenic progenitors from a normal iPS, so unaffected iPS. So we have now allogenic cell product that, if it's demonstrated safe in a first in human clinical trial, these cells can be used theoretically for different types of muscular dystrophy in different patients. So this is very exciting right now for us.